The Helicoverpa budworm moth is one of the worst global pests, threatening over 78 billion USD in total, as the target of most pesticides used in China, India, and a major fraction in South and North America, Europe, and Africa. A costly crop killer that has evolved resistances to almost all conventional chemical and biological treatments, including types of DDT and Bt bacteria. The advent of a more holistic ecological understanding coalesces with technological improvements to both articulate the current and near future populations' capabilities across Earth and exploit their physiological weaknesses in a precise, practicable, environmentally sustainable way. By distilling hundreds of research papers and grower accounts across several decades and dozens of countries, the best strategy can be deployed for specific contexts and regional dynamics. Budworms are so successful because they are hyperadaptive, neutralize the plant immune system at many levels, and have done so as a distinct lineage for around 10 million years. About 1.5 million years ago, a species of moth we now know as Helicoverpa armigera, the cotton bollworm moth, crossed the Atlantic Ocean more than 5,000 kilometers into the Americas from Africa, a highly adaptable insect belonging to the owlet moth family Noctuidae, and itself having developed over 7 million years ago in the Afro-Eurasian continent. Cotton bollworm larvae are accustomed to a wide selection of European and Asian host plants, especially grasses like barley, mallows like bull mallow and common mallow, legumes like chickpeas, and asters such as the European Michaelmas daisy. Despite the narrowly named association with cotton that researchers often use, cotton bollworm actually feed on hundreds of plant species, primarily due to a versatile set of detoxifying compounds produced in their saliva and intestinal tract, highly alkaline gut, internal microbiota co-opted from plants, and special immune tissues called fat bodies. Armed with the ability to quickly neutralize many common and unique plant defenses, as well as fly distances of 2,000 plus kilometers and deposit over 1,000 eggs per female, cotton bollworm established in this new territory in the mid-Pleistocene, a time of oscillating glaciation in the Americas and where the wild ancestors of many valued modern-day crops like tomato, tobacco, pepper, and maize developed. After establishing in its new region, the cotton bollworm moth began to adapt to the American ecoregions like the eastern temperate forests and Great Plains in North America, all the way south into the Central American Sierra Valleys and past the tropical wet forests into South America. Because the ancestral owlet moths from Eurasia, or more proximately Western Africa, developed on the flower buds and fruit of woody and herbaceous plants like the ancestors and current members of nettle, mulberry, buckthorn, legumes, and gourds, and even cannabis, this possibly pre-adapted the pioneering cotton bollworm's generalist physiology very well for the flora and fauna it would encounter in North America, quickly detecting and mitigating plant toxins in reproductive tissues by silencing communication between cells with a vitriolic cocktail of antitoxins and plant immunomodulators in saliva. Rapid slicing cuts hewn by serrated mandibles quickly remove small chunks of tissue while applying layers of saliva to the deepening wound tunnel, permanently cutting off downstream tissues from ever healing, which also serves to shelter the budworm from the elements and predators, a trait that develops in several relative owlet moth species. By chemically degrading and physically outrunning the defense response, budworms avoid the resulting increase in phytotoxin accumulation, trichome production, tissue silicification, and other intensified hardening effects, at least in the short term, against plant hosts that never encountered budworms yet and lack appropriate adaptations. With a caustic, scathingly alkaline gut pH of around 12, similar to that of calcium hydroxide or lime, and 1 magnitude below bleach, many microbes, enzymes, and other proteins, acids, waxes, and other structural compounds degrade and neutralize harmlessly and quickly pass, while some compounds may still go on to interfere with digestion of sugars and proteins, damage the gut membrane, or otherwise weaken the body, even lethally. Today, cultivated plants used for food are bred to develop sweeter or otherwise flavorfully stronger, larger, and more palatable leaves or fruit, which may require human care for maturation and defense against damage. For example, some landrace cannabis plants that have escaped cultivation for many generations develop a ditchweed phenotype of reduced floral size. Similarly, the reproductive floral and fruiting tissues of many wild plants, especially when unripe, are often small or not unnecessarily large, protected with toxins and dense tissues to disincentivize feeding and increase the success of reproduction. Selecting for a robust arsenal of penetrative tools, budworms evolved to bypass and process a food source itself evolved for a resilience on which survival of the next generation depends. Serious or even mild damage to flowers and fruits can restrict the success of reproduction, though crops in the last several millennia are often bred to increase the size and palatability of these structures, usually for culinary benefit, even if it may impair some innate pest defense traits. The flowers and fruits of grasses, in particular called caryopsis, typically very small with meager individual food value, are plentiful across swaths of plains and mountains in the Americas and Africa. As adult moths, cotton bollworms move with regional air currents and recognize both mates and potential hosts with keen eyes and chemical sensing antennae, which can both detect and associate certain volatile profiles with specific hosts, some of which may be more or less attractive for a variety of reasons. After more than 1 million years and several times as many generations, the cotton bollworm that established in the Americas fundamentally changed. This new American-adapted cotton bollworm population gained distinctive differences, such as a narrower band of gustatory and detoxification genes associated with a general drop to about 100 host plants spanning 29 different plant families, versus the West African origin cotton bollworm's impressive 300 plus hosts across 68 different plant families. A strong proclivity for the many verdurous American grasses, while retaining a still potent complement of detoxification, digestion, and chemosensory genes, make it an onerous herbivorous force known now as Helicoverpa, the corn earworm, for its critical preference of teosinte, which became the much larger, comparatively decadent, and more nutritious food source known as maize or corn that developed around what is now southern Mexico. During the 1.5 million year development of the corn earworm in the Americas, African cotton bollworm populations expanded and adapted to various regions like Western Europe, Central, Southern, and Southeast Asia, as well as Meganesia, like what is now Australia and surrounding lands. In fact, movement into the Meganesian region is somewhat of a homecoming, as recent genomic analyses show that the so-called native budworm, Helicoverpa punctigera, endemic to Australia, is the most basal ancestral species from which the entire Helicoverpa group descend evolutionarily. Meanwhile, breeding experiments conclude that native budworm, cotton bollworm, and corn earworm can interbreed and pass adaptive traits, so the potential historical and future long-time recombination between some or all of these species is likely to some degree. Earth's habitability fluctuated with the ice sheets over the past million years or so, as severely dusty storms percolated through a much drier landscape than today that locked away much of the water cycle encased in ice. The air temperature being about 6 degrees cooler than today, it was an environment that saw plant decline and environmental upheaval. Glaciation gains and losses, particularly northern Eurasia, created mosaics of isolated regions that periodically fused with surrounding areas and influenced the evolution of many surviving plant and animal species, including cannabis, through intermittent reproductive and ecological isolation. Stressors that might, as in other plants, incentivize reproductive traits, like monoecious flowering, that ensure seed formation with fewer individuals by establishing both reproductive flowers in a single plant. After the ice sheets retreated from their furthest reaches around 20,000 years ago, during what's called the last glacial maximum, several distinct cotton bollworm populations are recognized to have developed perhaps as early as 10,000 years ago, coinciding with the beginnings of human agriculture and cannabis domestication at various Eurasian sites within a general ecological warming, the beginning of the Holocene. Cotton bollworms in Central Asia's aridly polar Xinjiang region have clearly defined genetic differences, having adapted to the frigidly withering dry winds and freezingly gelid soils in which they developed as they diverged from southern and eastern Asian populations over several millennia. Sophisticated physiological analysis shows the perception of day length shortening or a reduction in photoperiod and decrease in temperature trigger in budworm moths the delivery of maternal signals to developing progeny, soon to be caterpillars, modulating gene expression to increase production and storage of the insect blood sugar equivalent trehalose, an osmotically stable sugar compared to the common plant sugar sucrose ingested, which has coincidental cryoprotectant properties. After sufficient development, larvae burrow into the soil to pupate within a soil-reinforced silken cocoon structure, and so for colder seasons insulates themselves during a metabolically slowed overwintering period before finishing development when it warms, emerging as moths. While the synthesis of trehalose is a normal byproduct of metabolizing digested plant sugars even for African origin cotton bollworms, the strong association of presently very cold conditions and the signal for even colder future conditions with increased provisioning of sugars important for simply surviving normal cold without food also had a secondary quasi-antifreeze effect after a certain threshold of trehalose was stored in their body. Those larvae that didn't accrue enough trehalose by winter in these severe Xinjiang conditions did not survive the sheer cold of minus 20 degrees Celsius. By supercharging this time sugar storage response to cold and darker autumnal periods, new areas and potential distribution channels became available, and may help in a tumultuous near future of predicted severe weather patterns. There are 4 major stages of the budworm life cycle: egg, larval, pupal, and adult. Understanding budworm capabilities also identifies their vulnerabilities. Adult moths have a somewhat bulky tan body, 14 to 18 millimeters in length, with a wingspan of 30 to 44 millimeters between wingtips. Inactive during the day, they typically start flying after twilight, feeding on nectar of various plants, mating for a few hours, after which females lay hundreds of small white dome-shaped eggs on suitable hosts in groups between 3 to 10, based on navigation by sight and scent. Adult budworm moths have been documented to prefer plant species on which they developed as caterpillars, making cannabis aromatic profiles a potential attractant, along with female pheromones that attract males. Pheromone traps allow for recording local population changes that inform preventative measures, but it may also be possible to exploit a reliance on volatile chemical signatures by introducing an aromatic superstimulus that attracts female budworm moths to a trap or disrupts their ability to discern the host from non-host over a general area, an area of research for several years that has yielded some breakthroughs. Female cotton bollworm moths in particular produce an antagonistic pheromone, cis-11-hexadecanol, that induces male avoidance for the few days the females are unable to conceive progeny and might interfere with reproduction if synthesized and applied strategically. Likewise, visual superstimuli such as ultraviolet light or wavelengths that mimic the green color of light reflected by host tissues can disrupt or direct moths in ways that reduce the chance of oviposition, especially where highly effective physical exclusion by walls or mesh screen is not really practicable. Preventing oviposition is one of the most potent mitigation strategies for budworms because it breaks the life cycle before any budworms can develop in the first place. Since larvae almost exclusively feed on floral tissue and rely on burrowing into the plant matter to shelter from environmental and biological hazards, immediately after hatching, no egg means no larva, as even mature larvae rarely move between individual plants unless all tissue has been exhausted. For this reason, preventative measures should focus on tracking pupal development to predict local emergence, reducing nearby adult development by targeting pupal chambers in the soil, adult movement into the crop space, and direct oviposition. In tropical environments with consistently hot temperatures like Central America and Southeast Asia, generations develop at a consecutive monthly frequency and can travel well over 1,000 kilometers on jet stream currents to temperate locations like those in North America and Eurasia. Temperate seasons typically impair pupal survival during winter above 40 degrees latitude, where soil freezes, but cold-tolerant populations from Xinjiang that survive -20°C may not be inhibited. Larvae that succeed in hatching spend a few hours consuming some surface tissue as well as a shell for vital nutritional and microbial input before boring into floral buds, pods, fruit, and other reproductive structures, which are highly nitrogenous. Within this limited threshold, larvae can be targeted, often with microbial or chemical agents that must be consumed by or applied to the body, but efficacy is dependent on timing and coverage, emphasizing the documentation of adult activity to inform the deployment of such preventive measures. Once larvae tunnel into the buds, management becomes a quagmire since contact-based products won't reach, systemic chemical products are undesirable, and even a hypothetical 100% kill rate results in caterpillar cadavers that are likely to foul floral tissue, particularly as the larva grows and defecates. Overwintering pupae will essentially stall development after a lower temperature threshold is reached, and die if temperature drops much lower, benefiting growers in areas where ground freezing conditions manifest, though, as mentioned, exceptionally cold-tolerant populations have been recorded surviving -20°C or -4°F, mainly in China, due to relatively recent adaptations in stored sugar reserves, which act as a cryoprotectant. Upon warmer days, development resumes, and depending strongly on the warmth of the ground, moths will eclose or hatch from their pupal casing, first at the earliest warming reservoir sites, followed by successive generational dispersion overlapping with regions that may still have dormant moths. This is because budworm moths can travel distances of 750 kilometers, mostly on their own power, or as much as 2,000 kilometers with often commonly adequate wind assistance in most parts of the world, upon globally connected airstreams called flyways. Active at night, adults take around a couple days to sexually mature and may mate before or after long-distance movement, with males attracted to pheromone compounds like Z11-hexadecanol and Z9-hexadecanol, and immature females producing that antagonist, cis-11-hexadecanol, to inhibit male non-optimal mating, substances and reactions that may be exploitable. Females can lay between a few hundred and around 2,000 eggs, but often about 1,000, either near their larval developmental area or after significant distance, influenced by factors such as larval experience, host plant perception, temperature, and nectar resources. Females have been recorded seeking hosts of the same species on which they developed as larvae, which may imply cannabis-specialized populations have been active for a while, perhaps centuries in the Americas and millennia throughout domestication in Africa and Eurasia, or even millions of years in wild prehistorical populations. Adult moths bereft of adequate food and water sources when traveling, such as in arid deserts or across seas and oceans, will be impaired and may fail to reach enough or any adequate hosts to deposit eggs, despite their wide host range. Even successful treks may not result in persistent establishment. Like other owlet moth larvae, budworms cannibalize others of their species, particularly later in development when food is scarce as a way to ensure access. Other pressures like birds and bats can thin adult populations, while parasitoid wasps and flies, specialized bacteria, fungi, and viruses target egg and larval stages to various degrees, degrading effective colonization. The total life cycle from egg to adult can complete within 25 days at 25 degrees Celsius, which is common in Central America, where around 11 generations per year is normal, whereas colder regions in and around Canada may delay local generational completion to 60 days or so, a developmental time of more than double. At their 5th stage of maturity, budworm larvae react to changes in light and temperature naturally at the cold end of the temperate season and will seek shelter, typically burrowing into soil about 2.5 centimeters or 6 inches, wrapping themselves in an insulated silken cocoon in which they will pupate and develop further. Pupae will die within soil that is too cold when overwintering, but can remain viable as far as 45 degrees north latitude, developing into adults much later than southern populations, though this may change with the introductions of cold-resistant populations. Although hot conditions are generally beneficial for insects, air temperatures much in excess of 30 degrees Celsius or 86 degrees Fahrenheit and as high as 37 degrees Celsius or 98.6 degrees Fahrenheit can inflict heat stress that, without mitigation, causes larvae to oversummer in a state of torpor at a reduced developmental rate, interfering with some physiological processes in particularly hot conditions. As has been quantified in budworm populations internationally, generational changes in physical attributes play a major role in survivability, which are influenced in larvae by access to ample host biomass, mainly floral and fruiting tissues of variously suitable plants. Some plant species or specific populations for various reasons facilitate or inhibit development, primarily the assimilation of nutrients, mutualistic microbiota, and ultimately mass, a sufficient amount of which is necessary to metamorphize, and only after surviving environmental and biological hazards. Starting off a few millimeters in length, cotton bollworm and corn earworm consume very tiny amounts of reproductive tissue, almost unnoticeable, while they bore and shelter. As the larvae grow, wounds on remaining matter fester with pathogenic infection. The majority of damage is met out in the final couple larval stages, which require the most front-loading of nutrition, making early detection vital for preventing the most damage and therefore yield loss, as well as targeting the weakest stage at its most vulnerable, arguably its only vulnerable moment, the interim between egg and tunneling, which can be as little as a few days, making strategic management actions just before seasonal peaks in spring, summer, and autumn paramount. Adults with larger abdomens appear to be correlated with increased metabolic reserves for longer flight and increased fecundity, as appraised in Chinese populations around the Bohai Strait and elsewhere. Larger thoraxes and forewings are associated with more efficient flight dynamics and increased endurance. These physical traits change throughout and between seasons of activity, affected by nutritional and genetic factors. While not typically feasible to assess in the field, such variation is certainly happening and may explain variable observations of budworm population or severity, and such physical adaptations enable future capability breakthroughs in movement, reproduction, and survivability, all of which make for a more odious pest. Decades of insect monitoring radar scans, catchnet and searchlight traps, and other sampling techniques show insect movement within the lower atmosphere of around 2 kilometers can be amazingly immense. In a 600 by 1,000 square kilometer region encompassing the North China Plain and East China Plain, which is the biggest food production area of China, one of the densest agricultural populations in the world, annual nocturnal aerial insect movement reaches numbers of 9.3 trillion individual insects, a combined biomass of 15,000 tons and 15.5 billion insects per kilometer, of mainly moths, piercing mouthpart insects like aphids and flies. This 600,000 square kilometer region is a vital section of an even more massive flyway that connects general bioflow between Indochina, Northeast China, the Korean Peninsula, and Japanese archipelago, and similarly vast channels exist elsewhere on Earth. Insects often alight with warm, fast-moving airstreams associated with the formation of low-level nocturnal jets at a few hundred meters altitude and may stratify at various higher levels, attaining wind-assisted ground speeds 2 to 4 times their unassisted capability, as they assess cues for host plant or mate location. Disrupting moth detection of crops through integrated physical or chemical means could significantly lower overall budworm incidence before more direct options are deployed. Cotton bollworm is an eminent presence across Africa and Eurasia, and with several unique populations adapted to environmental, chemical, and biological hazards present, as well as a significant proclivity for agricultural import and export, establishment abroad is plausible, particularly in proximity with the related corn earworm. To summate, ramifications of intercontinental genetic admixture are profound, as Central and South American budworm populations are exposed to different pesticides, many being banned in the USA like paraquat, atrazine, and thiamethosam, as well as safer alternatives found elsewhere like Bacillus thuringiensis strain Kerstaki and their very narrow-spectrum protein toxins, highly popular with medicinal or environmentally conscientious plant cultivation. These chemical pressures, combined with the torrid vibrancy of consecutive tropical generations and hybridization with cotton bollworm populations, increases the intensity of selection for further toxin, environmental, and microbial resistance. Currently, there are cotton bollworm populations resistant even to DDT, and common pyrethroids such as in Brazil, where products banned elsewhere such as the USA and EU are often applied, and corn earworm populations resistant to biocontrols like the commercially available and naturally occurring Helicoverpa armigera nucleopolyhedrovirus that kills only budworm larvae have been documented in various countries. Simultaneously, cold-resistant budworm populations that have been detected in Xinjiang, China, surviving freezing temperatures, and pesticide-resistant budworms in East China due to exposure and subsequent selection for larvae that express detoxification enzymes many times more abundantly and diversely than their weaker kin. These chemical, biological, and environmental resistance traits could be, and some cases already are, introduced to global populations, which would decrease the effect of several tools and conditions cultivators rely on for crop success against the most prolific pest of the most important staple foods. As of 2024, the global budworm population has established in most agriculturally important countries, with corn earworm and cotton bollworm sometimes overlapping together or with other Helicoverpa species. Global dynamics have been documented and unique populations analyzed, giving a holistic battle map growers across Earth can use to understand their specific regional environmental context and how it relates to their current and predicted future budworm capabilities for which to prepare. In 2005, hybridization between cotton bollworm and corn earworm was detected in Riverland, New York in North America. This may not be the only hybridization event since corn earworm diverged from cotton bollworm more than 1 million years ago, but it is the earliest documented case in scientific literature and implies that cotton bollworm populations are, to some degree, active in the Americas and have been for at least 2 decades, as of 2024. Although hybridization is well documented between corn earworm and cotton bollworm, there are a few intermediate reproductive barriers, mainly that there is enough dissimilarity between male and female physiology that complications and death can result more frequently from coupling than with their own species, and that pheromone composition can confound mate attraction. Of the several species in this genus, Helicoverpa, one species, Helicoverpa asulta, the oriental tobacco moth, has also been shown to hybridize with Helicoverpa armigera, the cotton bollworm moth, but only under controlled rearing conditions and never in field sample genetic tests, though its unique resistance towards irritating capsaicin, neurotoxic nicotine, and paralytic indoxacarb would be another defensive layer contributed to the collective budworm genetic repertoire. Regardless, beneficial trait admixture is a major concern for what is already the worst pest of so many staple crops globally, and the tropical regions of Central and South America represent a constant threat of budworm development to adjacent temperate areas that are major agricultural hubs, such as the North American Corn Belt. Influx across the Mexican border with the United States has been documented, as has several across the Caribbean Sea, Gulf of Mexico, and in fresh produce from international trade sampled at coastal ports, for which there have been hundreds intercepted in inspections. Northward, spring and summer migration of Central American populations into Southeast USA has the potential to introduce several resistances identified from not only Central and South America, but Africa, Asia, and Europe, for example, through trade routes. Populations can mix and propagate across the USA, where new traits may incubate and expand within North America before moving southward again or through international sea and air trade. According to Commonwealth Agricultural Bureau International, or CABI, an agricultural and environmental nonprofit organization founded in 1910 that, among other things, tracks pest research, the cotton earworm population that annually colonizes across the USA is composed of established populations and relatively continuous generations from Central American populations that technically reach far into South America. More immediate sites of maize and other relevant crop regions include Sinaloa and Jalisco, which are major concentrations of corn and other plants corn earworms consume, like tobacco, sorghum, tomato, pepper, sunflower, soybean, and cucurbits. These populations mix with those in the coastal chaparral and central California Valley, which then expand and mix into northern populations in the Great Plains' famous corn belt like Iowa and Nebraska, and eastern populations in the middle Atlantic coastal plains of Virginia and the Carolinas from March and April to October. Mexican ecoregions of the Sinaloa coastal plains and Sonora hills and canyons in the tropical dry forest region offer the most concentrated maize and related crops for budworms south of the U.S. border and may be an important breeding zone and vector route. Jalisco's southern semi-arid highlands, like the hills and interior plain region and transversal neovolcanic area of the temperate Sierras, are a warm transitionary space throughout the year, which may increase the intensity and mixture of adapted populations, establishing through sea trade from Asia, especially China, and land travel from South America, primarily Brazil. Feral and outdoor or greenhouse cultivated cannabis, including hemp, throughout the southern and midwestern area of the USA may become hosts for migratory budworms, and these regions could be the first impacted by budworms adapted to cannabis hosts or resist the narrow selection of approved biocontrols like Bacillus thuringiensis or BT. Areas where outdoor cultivation of primarily hemp has increased and decreased over time, especially since strong prohibition has waned and the 2018 Farm Bill became active. For the last few years, hemp acreage in the USA has been declining, but invigorated interest in textile, livestock feed, seed protein, and oil may change that if infrastructural support expands. In 2013, the first official but second actual documentation of modern cotton bollworm in the Americas due to reevaluation of genetic samples was detected in Mato in the areas of Brazil. Advanced genetic analysis identified the invasive cotton bollworms as an African population. Continentally, Africa is environmentally diverse, and several countries have experimented with cannabis cultivation as well as increased global trade, though biosecurity issues can facilitate establishment and expansion. In the last decade, since 2015 or so, sequenced budworm populations have had highly diverse backgrounds from various countries, confirming recent expansion. In Africa, growing ebullient trade and assiduous industrial activity increase and expand agricultural production as the same native cotton bollworm populations that seeded the American continents and eventually became the more specialized corn earworm, continue to plague much of the origin region, where they are liable to integrate with cold-adapted moths from East Asia and toxin-adapted moths from North and South America through commercial routes and some geographic flows. At its southern tip of South Africa, maize production for 2023 reached record levels in Northwest, Mpumalanga, and Free State, places of and near the high-veld grasslands where farming, industry, and population are concentrated. Budworm moths endemic to the area can easily travel in a southwestern direction on the wind currents, keeping to the greener zones on either side of the Great Escarpment, reaching into the Cape Mountains. Nearby Madagascar off the eastern coast may represent a likely vector both from international trade and stormy conditions, which can facilitate an aerial crossing that is already well within documented individual budworm ranges of more than 1,000 kilometers. Rainfall estimations predict southern Africa could become more arid further in the future near 2100, while the southwest may receive less rain and the southeast may receive mildly more within the next several years. Budworm travel between northern and southern Africa may be severely inhibited by the Congo air boundary, a combination of high peaks and valleys across the tectonic disruption known as the East African Rift System, which channels and collects torrential rain and tempestuous winds, isolating them centrally and disallowing the bulk of the storm movement, especially southward. Because budworms often ride local jet streams at altitudes as high as 1,500 meters in order to reach such formidable lifetime distances within the span of about a month, the wind and rain from storms severely disrupts flight on its own, with the steep vertical inclines that box in the storms adding another major physical barrier. Since budworm moths often mate within a few days of developing and just before long-distance flight, rain, wind, and high-terrain features may contribute to earlier adult death instead of localized reproduction, while other aerial factors like haze, fog, and smog, or other air conditions may disrupt moth perception and other biological processes like scent perception. Ethiopia in northeastern Africa exists within the estimated origin zone of cotton bollworm. Its mountain highlands and tropical monsoon regions inflict strong boundaries for moth flight, creating pockets of locally adapted subpopulations, while direct human transport from the eastern border with Somalia, as well as that from Mexico and China, perforates these environmental confines sharply and are expected to flourish with historically high collaboration in the coming years, which could mean the spread of BT and cold-resistant populations not only in Ethiopia, but associated trade partners. Sampled Ethiopian populations of cotton bollworm during the 2018 cropping season were associated with many countries, particularly Brazil, Australia, and India, with high genetic diversity implying recent establishment. Brazil, in particular, represents a potential source of budworms with deft toxin-defeating capabilities against both pesticides and natural plant defenses, threatening an already dyspeptic, indurated agricultural context with few practicable options. Ethiopia's Oromia region is an agricultural center, especially for khat, coffee, tomato, and chickpea, with the latter two crops increasing steadily. The budworm population sampled in this specific area showed signs of low genetic diversity, meaning populations were generally stable and local, with a recent admixture of populations from South Africa, Zimbabwe, Kenya, and some European countries like France and Spain. In the 6 or so years since sampling, invasion throughout Ethiopia broadly and ag sites like Oromia has likely continued, and speculatively, the close proximity to Somalia and the Strait of Hormuz may facilitate budworm population movement primarily by sea trade, but also northward into the Mediterranean and Central Asia, like happens for many other insects, fungi, and other organisms. Western Asia, encompassing the Arabian Peninsula and neighboring countries, is a mainly arid mountainous region replete with human historical importance going back many millennia, with several crops being domesticated or improved here, like cannabis, wheat, barley, and chickpea. Agricultural trade and migration by land and sea in modernity could help budworms bypass the more inhospitable climb and locate sparser ag land and pockets of alternative hosts, while innovations directed toward resource challenges must make progress under this context, straining a worn and literally ancient logistical hub that could fray into an unfettered catastrophe as temperature and droughts rise. A proportion of budworm moths entering this area by air may be slowed or totally expend metabolic resources like sugars, mainly glycogen and trehalose, and fats such as triacylglycerol before laying eggs on a preferred or suitable host. But with its triple proximity to Africa, Asia, and Europe, low biosecurity and monitoring capability, a plague of megaworm hybrids with various resistance traits from these regions could mate with native arid-adapted populations and, of those that survive, rapidly sweep through without much forewarning. Such a contingency would contribute to a humanitarian crisis and resource insecurity in this and neighboring regions already underway, such as in parts of Africa. Feral and landrace cannabis in and around these regions has a tumultuous history, and budworm prominence is another potential threat to vital genetic lineages that may have pest and environmentally adapted traits that should be preserved and protected from external threats. Iran's proximity and variable environment encapsulates this context. Severe deserts in the east, like the southeastern Lut Desert that reaches an infernal 70.5 degrees Celsius on the Gandambarian Plateau, comparatively crisp and refreshing Hyrcanian forests that border the Caspian Sea to the north, that are themselves isolated by the Alborz Mountains and a double-thick vertiginous wall composed of the central Iranian and Zagros mountain ranges for much of the west that is practically impenetrable. Most of the world's saffron and 15% of the world's pistachios as of 2023 are produced in Iran, though water scarcity, poor soil quality, and severe logistical stressors exacerbate an already difficult circumstance, much like other parts of the Arabian Peninsula. Most uniquely, a link between agricultural subsidization and volatility of oil prices. The province of Golistan, a name meaning land of flowers, in the north is a site of agricultural research and production, mainly cereals, though capricious flooding and fires impairs developments perennially. In a budworm moth forecasting test, land surface temperature from satellite data fused with known cumulative changes in surface temperature unique to the budworm biology correctly anticipated first emergence time and location of overwintering cotton bollworm in 2020, particularly strong in the intersection between northern Gorgon County, western Gonbad Cavus, and the eastern tip of Turkoman, near the foot of the Alborz Mountains. Because different places will warm at variable intensities across a 24-hour period and over a minimal threshold for budworms to develop, which the study pegged at around 10 degrees Celsius, such locally precise predictive analyses are crucial for the most efficient and sustainable preventative responses, such as the application of biocontrols, pheromone traps, and culling of potential hosts and pupae burrowed in the soil on property right before moth emergence. Thermal data from meteorological stations was less accurate than land surface temperature taken by a MODIS sensor on a Terra satellite, but in a pinch, or when reinforced with LST info, it worked well and could be a potent communal resource for cultivation in most environments, as has been investigated recently. In Akala County, for example, models based on land surface temperature predicted 50% of budworm moth emergence after accumulated degree days of about 255, which is when the combined mean daily temperature minus the minimal threshold of about 9.75 Celsius reaches 255. If, hypothetically, the mean daily temperature is 25 degrees Celsius, then 255 degree days would accrue in about 10 days. In practice, this starts to matter sometime at the end of winter and beginning of spring when the mean daily temperature rises past that minimal threshold, 10 degrees Celsius or so, whereas in tropical locations, development is constant. More specifically, since budworm pupae rest insulated in a silken cocoon underneath centimeters of soil that may or may not have snow, shade, or other complicating factors, mean surface temperature at ground level better represents pupal experience than air temperature, and such climate information is often freely available. CAPI states that 1 hectare, or 10,000 kilometers squared, of U.S. cornfield can produce 50,000 corn earworm moths in spite of control measures. The most intense corn production counties in states like Iowa, Minnesota, Nebraska, Kansas, and Illinois can reach 1,500 hectares individually, which would imply potential numbers in the hundreds of millions or billions per county annually from corn alone. This assessment would make much of the Midwest a high-threat zone and may be a concentration point for regional hybridization. With a likely minimal dispersal range of 750 kilometers, the epicenter of the Corn Belt reaches most of the Midwest within 1 generation. At a plausible max range of 2,000 kilometers, the Central Corn Belt budworm generation reaches practically all of North America and even into Mexico. Research also shows that corn earworm moth will seek plants similar in aromatic profile to those on which it developed, and with historical feral cannabis populations of so-called ditchweed present in the Midwest and Southeast, as well as areas in Mexico and Central America, it may be the case that cannabis-oriented budworm populations could have existed in North America as early as the first cannabis plots in U.S. history, surviving amongst feral groups that escaped cultivation and more recently expanding in response to increased outdoor and greenhouse cultivation. To give an illustrative example, there are some speculative megaworm populations that might be found in the future based on some of the characteristics that we're familiar with now. For example, perhaps the USA will develop the Great Plains budworm, a hybrid of populations from USA, Mexico, Brazil, and eastern China. The 8 to 12% greater average adult mass and larger wingspan increases typical maximum range to 3,000 kilometers, and maximum reproduction documented to 2,800 under ideal conditions. Larvae are not visually distinguishable, but strongly resist a wide assortment of chemical agents, most BT strains, and have a cold hardiness that greatly increases their survival above the 42nd parallel, a former limitation. Reliable reproduction with cotton bollworm and corn earworm maintains fresh adaptations. In the People's Republic of China, perhaps the small wing budworm Xiaoyiyachong would develop. First documented in the Bohe Strait, as well as several East African countries and India, a shorter, rounded forewing and 13% larger thorax muscles are associated with adaptation to short distance travel and mountainous regions. Larval preference for legumes like pigeon pea and similarly protein-rich crops has been confirmed in choice tests. A strain of Enterobacter called ZE7 interferes with the colonization of baculovirus virulence in budworms when tested in other groups, but is found only in these populations, implying a strong adaptive significance or maternal link of the bacteria. Perhaps in Egypt the brown budworm would develop. Darker melanization of larval cuticle associated with increased resistance to solar radiation, a need brought about from the lack of adequate food in arid terrain, which requires quickly moving into new bud sites, sometimes between individual plants across the hot ground. Several genes associated with oxidative stress reduction mitigate high temperature damage in mature larval stages, which are not found in other Helicoverpa populations. Larvae readily cannibalize other budworms of the same size or smaller, as well as other caterpillars, representing a rare case of adaptive carnivory in a primarily herbivorous moth species. The cotton bollworm microbiome is greatly influenced by the phytoplankton microbiome, colonizing the epidermis and endophytic microbes residing in consumed tissues, as well as the probiotic eggs larvae eat immediately after hatching that contain many important microbiota. Pathogens like the Helicoverpa nucleopolyhedrovirus, or Beauveria bassiana the fungus, and similar entomopathogenic fungi that associate with plant tissues naturally colonize the larvae, usually lethally, but some microbes like Enterococcus and Enterobacter have been shown to help neutralize toxins of Bacillus thuringiensis by increasing gut alkalinity past structural tolerances. In fact, these two groups are some of the most dominant bacteria in moth species generally. Other bacteria found in budworms at various stages include Commensalibacter, Chryseobacterium, Acinetobacter, Lactococcus, Apibacter, Bartonella, Acea, Orbus, Disconomonas, Organella, Pseudomonas, and even some Rhizobiales, known for establishing mutualistic nitrogen-fixing relationships with plants. Based on functional analyses, many metabolic and signaling effects were associated with various microbes. Solid frass droppings can contain or facilitate the development of local plant pathogenic microbes, particularly in the proximity of fresh wounds, which they are highly likely to contact as the caterpillar bores through the floral mass. This and the strong likelihood of a cadaver resulting from even successful treatment post-tunneling is a unique context of boring caterpillars and crops, even if systemic products might be compatible with cultivation practice. Since a dead caterpillar, especially a larger stage, will rot and fester in bud tissue if killed inside the structure, early detection and prevention are important. For the most ideal outcome, options must be practical for a cultivation context to prevent larval tunneling. With the holistic, multi-domain consideration of budworm ecology, capability, and vulnerability, strategic decisions informed by empirical evidence can be made with greater assurance. Cultivation contexts like geographic location, scale, and available resources is the ultimate doctrinal fulcrum by which a plan can be enacted. Generally, budworms are best prevented, particularly by severing the adult-egg connection, by denying host plant access, typically by using a physical barrier or layered microbial application. But there are other factors. Prevention strategy for budworms in cannabis specifically must be able to integrate with other pest strategies into a synergistic pest management system and account for environmental and logistical context like known seasonal pest activity, both current and anticipated, local topography, harvest dynamics, resource accessibility, and other cultivation idiosyncrasies. A focus on budworms should not disregard other complicating factors, which is a strategic mistake that is commonly facilitated by lack of contextual information, such as the presence of new or adapted pests, or higher populations, externalities of great adjuration like resource austerity, or critical extenuating circumstances, such as a natural disaster or personnel changes. Acquiring relevant contextual information and requisite resources for the cultivation context are prerequisites for efficient prevention strategy. Targeting physical constraints like the size of moths, the structure of their larval bodies, or physiological limitations can be extremely cost-effective, conceptually simple, and resist adaptation by exploiting fundamental attributes that can't be easily changed. Since moths must make contact with floral tissues to deploy eggs, which then become larvae, a potent passive countermeasure involves the physical exclusion of flowering plants, such as with a fine mesh screen or insect netting, though this is more resource intensive to accomplish at greater scale and a common aspect of greenhouses. Similarly, multiple physical partitions that surround the cultivation space, such as structural walls, in addition to natural or artificial windbreaks, disrupt moth movement and perception of host signals, particularly in flat, windswept landscapes. Behavioral ecology informs vulnerabilities like the reliable reaction to certain environments, volatile chemicals, light, and general activity. Pheromone traps that attract male moths can be effective for timing local population activity, informing the deployment of reactive measures with as little operational waste as possible. Ultraviolet light traps may kill budworm moths, but they do so by attracting them, which may be a double-edged sword that typically comes with plenty of non-target insect bykill, many of which can be environmentally important as food and pollinators, threatened species, or even budworm enemies. Locations that experience cold winters that induce overwintering create an operational reprieve of a few months that can be used to destroy potential pupal reservoirs underground and nearby host plants, reinforce physical barriers, and make other changes before temperatures rise. Areas with ground temperatures that approach or exceed freezing are more resistant to non-adapted budworms. Similarly, locations that experience high air temperatures much over 25 or 30 degrees Celsius with low vegetation can be suboptimal for long periods, and exposed moths in austere conditions may have difficulty mating, flying, and finding hosts, making some arid environments natural barriers. Acclivitous mountain terrain, precipitation, and various storms also contribute to moth attrition and suppressed range, so growing in or around these conditions may have passive protective effects. Conversely, budworm moths benefit from environments that are flat and consistently warm, exemplified by plains and tropical regions respectively. Ample floral hosts for larvae to shelter in and moths to drink from facilitates regional colonization. Reliable, straightforward wind currents disperse moths across continental distances easily, so while zones of proportionally productive biomass like plains and forests and concentrations of agricultural land or private gardens can sustain local populations, unmitigated regional influx overlaps with these areas such that the incline, decline, and adaptation of populations resulting from dynamics far away and seemingly removed from a cultivation area can and do reach expeditiously. Comprehensive preparations must account for this ever-present swarming context that can substantially change between seasons for unpredictable reasons. Emphasizing location choice and structural safeguards that reduce the incidence of egg laying and subsequent larval development in the first place, neutralizing the costly interaction since preventative applications, crop scouting, and resulting dead and potentially fouling larvae that may still be in the floral tissue from successful management is still logistically burdensome to track. Prevention measures in cannabis still might encompass a biological package of natural enemies such as Bt bacteria, Beauveria fungi, viruses, and egg-killing Trichogramma parasitoid wasps, as many chemical products are undesirable in flower because they may have a long residence time, strong odor, or simply disrupt floral or trichome characteristics, let alone the propensity for specifically systemic chemistries to be highly toxic to people and environment, and the chronic feckless ambivalence with which such products are applied. Parasitic wasps and flies, like other biocontrols, perform better when provisioned with alternative hosts in the nearby area, and water or nectar resources like the small florets produced by various mint and umbel species like lemon balm and carrot respectively. Sweet alyssum is a popular and venerable choice, often found in research and field conditions, and it should be noted that yellow and white are considered particularly attractive to the biocontrols. Banker plants and similar natural enemy field reservoirs can be established on site in interspersed, adjacent, contiguous, or piecemeal configurations to provide food and shelter. Plants endemic to the location or with similar adaptive traits can make management easier, but care should be taken to choose plants less likely to support prevalent cannabis pests and evaluate banker plants for symptoms therein, such as thrips, whitefly, and spider mites. Banker systems are reasonable only in contexts where they decrease logistical costs. Ambient populations of natural budworm enemies like predatory or parasitic insects, documented in some field research investigated since the early 1900s, have not been found to be reliably protective. Seemingly, this is because natural enemy populations within an open area follow host population spikes and depressions closely, and will generally lag behind this influence like overwintering generations and sudden migratory swarms, an ecological phenomenon called hysteresis. Even compensating with concentrated inundative applications of, for example, parasitoid wasps made in anticipation of budworm presence can still result in yield losses by themselves, making resource provisioning and other support vital for maximal effect. Spiders, predatory stink bugs, and similar generalists show some beneficial effect in controlled and field conditions, especially if allowed to establish without strong pesticide exposure. Insectivorous bats and birds may benefit nearby growers by feeding on many moths in the vicinity. Microbial enemies as well as mutualists exist naturally on and in plant tissue, and biasing the populations towards budworm enemies is one of the most popular and effective biological options. Although necrotrophic fungi that rot tissues like botrytis and fusarium often exploit wounds left by tunneling behavior, there are some strains of such species that deviate significantly from their more infamous pathogenic relatives. Several species of fusarium associated with soil and plant hosts are capable of colonizing and within 24 hours killing budworms with insecticidal compounds like fusarin C, moniliformin, and eniatin peptides like buvericin, along with many other enzymatic factors. In the soil of a suburban green area in Patras Archaea, Greece, an isolate of Fusarium solani called Delta 99b was found to be particularly toxic to budworm hosts. A strain of Serratia marsans, a highly prevalent bacteria even found in the human microbiome, has been shown to break down the budworm exoskeleton by degrading chitin, and may inflict other damage that may make it or a similar strain a potent agent of biocontrol. Commercially available biocontrols are species that have been studied for reliably effective management traits for certain pests and reared in conditions that maximize them. Beauveria bassiana, for which buvericin is named, is one of several common entomopathogenic fungi that have worked well in field use for many insects, and several strains of Bacillus thuringiensis like Kerstaki and Aizawi that produce toxic protein crystals that only activate in a sufficiently base environment, like the caterpillar gut, and are destroyed by acidic environments. In budworms, these proteins, like cry, form structures that puncture gut wall cells, shortly leading to sepsis, particularly for younger larvae. Timing from the earliest onset ensures contact at just about the only appropriate stage, while tunnels sheltering the larvae in older individuals resist these substances much better. Baculoviruses like Helicoverpa armigera nucleopolyhedrovirus, or HERE NPV, are exceedingly commonplace in agricultural and forested areas, with viral particles recorded in concentrations of 10,000 to 600,000 virions per gram in both forest and crop leaves, with soils acting as an immense viral reservoir that can allow windborne movement back into foliage. Commercial strains are precise and often species-specific, and while budworms can develop resistance to natural infection and even pass the virus to progeny in certain cases, exposure to new strains and sufficient inoculative pressure enervates this advantage. Targeting bacteria locally in the gut or other microbes picked up on the exterior phytoplane or terrestrial topsoil could severely impair budworm physiology at egg, larval, pupal, and adult stages. Essentially, there are a few avenues to consider: inoculating the plant with budworm antagonistic microbes, reducing the presence of budworm beneficial microbes by chemical or microbial applications, and less directly by priming plant defenses and growth responses before budworm presence. Endophytic microbes that can subsist in plant tissues, especially directly parasitic fungi like Beauveria and Aizaria, might become a more resilient and cost-effective countermeasure with the right inoculation technique and symbiotic optimization. Mutualistic microbes that have priming or other indirect effects that bolster a range of physiological processes can have significant influence on a host plant's response. With accurate localized data for both springtime emergence and peak moth activity, which is often sometime around late summer in temperate climates, populations can be anticipated and germination timing altered like in other crops in order to avoid flowering during this period as much as possible, and biocontrol applications can be implemented right before presence mounts, maximizing prevention efficacy. Sensor and data collection options for growers have substantially increased in the last several decades, and the fusion of environmental, ecological, and operational data from various sources crystallizes disparate and overlooked information into a coherent representation of past, present, and future predictive pest forecast analysis and modeling in the cultivation area. Quantifying the spatial distribution and intensity of budworms over time across the crop is most important as an objective measurement of context, specifically change, needed for informed action. Wormed buds can appear in clusters of nearby plants and may be closer to the perimeter or entrance of structures as represented in spatial tracking, so scouting or applying preventive measures with a budworm moth behavior bias can increase management speed and success. International efforts to collect and process massive amounts of such broad-scale information and make it available and easy to understand should eliminate ambiguity and relieve some burden, in addition to the development of novel options. Like certain benefits from social media and communication of effective practices, such a connection could be a force multiplier whereby the combined benefit of each grower's hardened defenses reinforces nearby growers like armor plating, and may stymie future and degrade current selection pressures related to toxin resistance by relaxing one-dimensional chemical applications for a multi-domain initiative. Since entomological documentation by Jakob Hubner 219 years ago in 1805, cotton bollworm continues to be extensively studied and researched, as well as locally relevant detection and management strategies are publicly available in many places to maximize results for individual growers. Manual scouting generally focuses on overt signs of damage, like floral necrosis, cottony white bud rot, and bract tunneling, as the white eggs are about 0.5 millimeter wide and often nestled in bract crevices, and the first instar larvae are not much longer, feeding externally for perhaps a few hours. Confirming presence upon the detection of symptomatic damage and recording it physically and digitally streamlines documentation for future review, and in cases where location is mapped, illustrates seasonal population changes over space and time, such as initial incidence, peak incidence, final incidence, and response to management. Pheromone traps that attract males can be deployed before flowering starts to gauge local activity, inform application of preventative measures, and contribute to baseline data for expected seasonal activity. For several years, efforts to identify and differentiate between budworm moth species using computerized vision models have yielded systems with precision as high as 95% and above, but practical implementation in the greater cyber-agricultural ecosystem has been insufficient and slow, with a lack of direct pest mitigating effect and the need for equipment like various cameras and software to constantly scan for threats. Perhaps for a specialty crop like cannabis, key installations at perimeter or entrances to greenhouses would be cost-effective and increase the robustness of scouting, particularly in the deep night and very early morning, when most scouts are not active, but moths are. Infrared scanning for moths may be more useful for nocturnal monitoring, like the Dutch PATS-C system and the associated anti-moth unmanned aerial vehicle, the PATS-X, a small drone that takes spatial information to seek and impact target moths using propellers. While disintegrated moth particles are not really desirable in sticky floral bud sites, interception outside or before flowering could be useful, as it has been in ornamental floriculture. Glue traps with mounted cameras for identification and programming for alerts, recommendations based on contextual data, and even automated replacement of the glue traps themselves could radically hasten the flow of information and speed of informed decision-making as soon as the first budworm adheres. Manually checking, collecting, evaluating, and replacing glue traps at regular intervals is often laborious and time-intensive, the information potentially days old at time of recording, whereas several automated glue traps deployed at specific intervals would characterize and log each encounter, showing incidents over time and space, illustrating behavioral and temporal patterns, and freeing up personnel for other important considerations. For most contexts where plants are exposed to the environment, this pest represents a high threat not only because a single larva can severely decrease the flower or fruit yield of an entire plant at almost any point in the reproductive timeline, but also the sheer volume of pinhead-sized eggs laid by a single adult, easily reaching several hundred or even 1,000 or so, with multiple eggs often deposited on a single plant in a crevice of the inflorescence, and early larval stages burrowing cryptically at first, showing signs after significant damage has already occurred. Budworm toxin resistance is not only persistent, but indurated with successive exposure to new plants, synthetic chemistries, and hypernatural toxin concentrations. However, there are plant defenses upon which populations are shown to develop suboptimally, and facilitating similar or improved natural traits in cannabis, targeting the same vulnerabilities is perhaps the most efficient option if the traits are validated and sufficiently robust in the face of budworm adaptation. Trait expression is complex, and studying specific pest resistance factors requires technical resources and verification, but speculatively, such traits could be anything from directly disruptive compounds like those in glandular trichomes and general tissues, physical structure like pointed non-glandular trichomes or silicofied cells that degrade feeding or rupture gut lining, or even improved signaling response that makes surging certain defenses more rapid on a physiological scale. More indirect traits could be related to something like bract density or time of flower onset and maturity. Research in this field for cannabis and many other crops is expected to continue at a sedulous persistence.